So in previous videos, you have learned that citric acid cycle is the main source of NADH and FADH2. The catabolism of carbohydrate, as well as the catabolism of lipids and proteins, feeds the citric acid cycle and produce even more molecules of NADH and FADH2. The final step in catabolism under aerobic condition is oxidative phosphorylation. So in this video, I will give you a quick overview of the fate of electrons during oxidative phosphorylation. During oxidative phosphorylation, NADH and FADH2 will give the electrons to be converted back to NAD+ and FAD. The electrons that are given move through multiple transport steps before they will reduce the final electron acceptor, oxygen, to form water. This exergonic process is coupled with ATP synthesis. Now, where does oxidative phosphorylation take place in eukaryotes? Remember that the citric acid cycle takes place in the mitochondria. Therefore, a large amount of NADH and FADH2 is produced inside mitochondria and cannot diffuse outside of mitochondria. And in fact, oxidative phosphorylation will also happen in mitochondria. Mitochondria are surrounded by a double membrane. The outer membrane is very permeable to small molecules and ions. The inner membrane falls into cristae and is impermeable to ions and small molecules. In fact, you need specific transporter to move small molecules and ions across the inner membrane. This inner membrane is also extremely rich in protein that form the electron transport chain, and in one enzyme that is responsible for the synthesis of ATP and is called the ATP synthase. Between the inner and the outer membrane of the mitochondria, you have the intermembrane space. The interior space of the mitochondria, called the mitochondrial matrix, is very rich in enzymes, in particular enzymes involved in the citric acid cycle, but also in the catabolism of fatty acids and amino acids. How does the electron transfer leads to ATP synthesis? The answer is based on the chemiosmotic theory that was proposed by Peter Mitchell in 1961. Electrons are transferred through a series of carrier molecules and proteins. The electron flow moves in a downhill exergonic direction, providing energy to power the uphill transport of proton across the inner membrane from the mitochondrial matrix to the intermembrane space. The free energy derived from the oxidation of fuel molecules and ultimately from the transfer of electrons through the electron transport chain is conserved as a transmembrane chemical potential called the proton motive force. This gradient of proton across the membrane is in fact an energy reservoir that is used to drive ATP synthesis. ATP is produced when the protons flow back into the mitochondrial matrix down a concentration gradient through the ATP synthase. The ATP synthase uses the energy harnessed from the proton flow to phosphorylate ADP and release ATP. Now let's go back to the origin of oxidative phosphorylation and focus on the delivery of electron to the electron transport chain. Where do these electrons come from? Well, we know they come from oxidation reaction taking place in a variety of catabolic pathways. These electrons are then funneled in one of few universal electron acceptors. For example, we've talked about NAD. NAD is one of these 4 universal electron acceptors. And NAD is, in fact, the nicotinamide adenine dinucleotide. NAD is reduced into NADH and a proton. This reduction is always catalyzed by one type of hydrogenase. The electron extraction during a catabolic reaction and the reduction of NADH are in fact coupled. In this reaction, the reduced substrate reacts with NAD+ and is oxidized in an oxidized substrate, and NAD+ is reduced in NADH plus H+. For example, in the citric acid cycle, malate is oxidized into oxaloacetate, and this reaction is coupled with the reduction of NAD+ into NADH plus a proton. And the reaction is catalyzed by the malate dehydrogenase. If we take a closer look at this reaction, we see that when malate is converted into oxaloacetate, malate will lose 2 hydrogens and 2 electrons. One of these hydrogen and the 2 electrons will be transferred to NAD+ to form NADH. The other hydrogen is released as a proton. NADH can now carry these 2 electrons to one of the components of the electron transport chain. There is another universal electron acceptor, NADP, that belongs to the same family of nicotinamide adenine. NADP is the nicotinamide adenine dinucleotide phosphate. So it is a phosphorylated version of NAD. The same thing will happen for NADP+. It will accept 1 protons and 2 electrons to form NADPH plus a proton.
What's the point of having both NAD+ and NADP+?
Well, your question is well-timed because I was ready to address this issue. In fact, NAD+ and NADP+ have 2 different functions. NADPH fuels electrons to anabolic reaction or biosynthetic reaction. NADPH is not directly involved in the synthesis of ATP. In contrast, NADH funnels its electron to the electron transport chain and is responsible for catabolic reactions and the synthesis of ATP. In fact, the dehydrogenases that are used by NAD+ are very often found in mitochondria, while the NADPH-linked dehydrogenases are usually found in the cytoplasm. This distribution contributes to the partial separation between anabolic pathways and catabolic pathways in eukaryotes. The second major type of universal electron acceptors belong to the family of Flavin molecules that are derived from vitamin B12. We have seen already an example, FAD, which is the Flavin adenine dinucleotide. FAD accepts 1 electron and 1 proton to form the semi-reduced FADH, and then will capture another electron and another proton to form the fully reduced FADH2. Another example of the universal electron acceptor that belongs to the Flavin family is FMN, the Flavin mononucleotide. In the same way, FMN will react with 1 electron and 1 proton to form the semi-reduced FMNH, and then will capture a second electron and proton to form FMNH2. So in conclusion, 3 of the universal electron acceptor that we have described, NAD+, FAD, and FMN, will provide metabolic flexibility and will deliver electrons at different points along the electron transport chain, as we'll see in a later video. In the next video, we'll talk about the proteins that accept electrons from the universal donors, and you will learn how this process provides a source of energy that drives ATP synthesis.